Welcome to the webinar today. This webinar is entitled Advancing the Culture and Clonogenic Analysis of Gastrointestinal Stem Cells. My name is Scott Schachtel, the product manager for stem cell and cell culture tools at R&D Systems, a Bio-Techne brand, and I'll be your moderator for today's event. I'd like to thank everyone for attending, and I'm very happy to welcome our featured speakers today. The first speaker is Dr. Frank McKeon, Director of the Somatic Stem Cell Center at the University of Houston in Texas. After the presentation, Dr. McKeon will be joined for a technical question and answer session by Dr. Xian, the Assistant Professor in the Center for Stem Cell and Regenerative Medicine at the University of Texas Health Science Center. This webinar is supported by Bio-Techne. Bio-Techne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tokris Bioscience, Protein Simple, and Advanced Cell Diagnostics to provide the scientific research community with a comprehensive and world-class product portfolio of reagents, media, extracellular matrices, small molecules, assays, and instruments that can support the generation and analysis of 3D cell culture models. Before moving into our featured presentation for the day, I would like to just take a few minutes to highlight a couple of tools that may improve your in vitro gastrointestinal model systems. The first is an exciting new platform for the generation of accessible and robust 3D gastrointestinal tissue, which we're calling MimEX GI. The MimEX GI is the first system available within the MimEX Tissue Model Systems product line and provides the media and reagents needed for isolating and expanding adult stem cells from the gastrointestinal system. An example of this is shown of a stem cell colony as shown in the bright field image on the upper left-hand corner of your screen there. The system also provides media for the differentiation of these stem cells into 3D tissue in vitro, as shown in the image on the right-hand side, showing immunocytochemistry of some differentiated tissue. The MimEX GI media and reagents are built from foundational work out of the labs of Frank McKeon and Xian. I'm very excited to have them here today speaking about how they're using their technology to widen our knowledge of gastrointestinal stem cells and their potential use in understanding disease mechanisms. The schematic here on the screen just describes the basics of how the MimEX GI reagents and media work in the platform. So adult gastrointestinal stem cells can be isolated from biopsy tissue or purchased directly from R&D Systems. And these cells are expanded in a traditional 6-well plate, pre-coated with culture basement membrane extract, and then seeded with MimEX irradiated fibroblasts. When cultured in these conditions with expansion media, the stem cells selectively proliferate. These are then harvested and plated in a high-density monolayer in a transwell insert system. Under this air-liquid interface and in combination with the MimEX differentiation media, the stem cells begin to differentiate up and into the 3D gastrointestinal tissue. So this system provides a number of benefits to researchers, including better accessibility to the apical epithelial surface of the 3D tissue, more control over tissue consistency, the acuity to generate region-specific tissues, as well as tissues from diseased donors, as well as clonal selectability, which lends itself to gene editing techniques. And Dr. McKeon and Dr. Xian will be able to elaborate on the science behind these features in their presentation. Last, Bio-Techne also offers a range of tools across the 3D and stem cell culture workflow, as illustrated on this slide. I just wanted to bring it to your attention that we're really trying to provide and be a resource for researchers so that they can come to find high-quality reagents and medias in one location, from proteins and small molecules to cell scaffoldings or media supplements. So really trying to be a resource for the research community in one location. A key resource for the 3D culture system is our organoid reagent guide webpage, which shows reagents and tools that can be used to generate different types of 3D culture systems, kind of organized in this tabular form. And the URL there is on the screen. So I suggest you visit there as you're trying to build or optimize out your 3D culture systems. Okay, so thank you for listening to the introduction there. Before handing over controls to our speaker today, I have just a few housekeeping items to address. Throughout the webinar, we invite you to submit any questions you have for our presenters using the Ask a Question box just below the presentation screen. These questions will be asked to the presenters directly following the presentation. And then in the upper right-hand corner of the screen, you will see a tab called Resources. In that tab, you'll find extra content that relates to today's webinar. Okay, and with that, I'd like to introduce our speaker for today, Dr. Frank McKeon. Dr. McKeon is the director of the Somatic Stem Cell Center at the University of Houston in Texas, as well as a professor in the University of Houston's Department of Biology and Biochemistry. He received his PhD in the lab of Dr. Mark Kirschner at the University of California in San Francisco. Dr. McKeon has developed novel stem cell cloning strategies to model the airway system and gastrointestinal tract. As mentioned above, these strategies are the driving basis for the transformative MimEX tissue model systems by R&D Systems. Dr. McKeon's continued work using adult stem cell-based models of epithelial organs is advancing our understanding of lung regeneration, the origins of highly aggressive upper and gastrointestinal tract diseases, and the complex biology around gastrointestinal stem cells. And with that, thank you very much, Frank, for joining us today, and I'd like to hand over the controls for the presentation to you.
Terrific. Thank you, Scott, for that kind introduction. I'm here with Xian, who I think people who know us know that we've shared a laboratory for the past decade, both in Singapore and now back in the U.S. We're quite thrilled to make this, together with Bio-Techne, this technology available. I think there's a number of barriers to entry regarding this technology, and I think Bio-Techne is doing their best to limit those barriers and to make it available. And so with the more the upfront work, I think people will see there's a number of significant and perhaps transformative advantages of the technology. So I'm going to run through this. It's going to be somewhat of a rambling conversation here, but please look forward to your questions, and while we'll handle those as they come in. So let's just go to the first slide. So this is technology for cloning adult columnar epithelial stem cells. And this is not new technology. In fact, it's older than the generation of embryonic stem cells. It largely follows on Howard Green's epidermal stem cell cloning. And the key there was that he was able to maintain stem cells in their most immature and clonogenic state in a 2D assembly. So this is not organoids. So they're not being maintained as organoids. And yet, under different conditions, you can differentiate these at will. So we're into the realm of adult stem cells, and there's just a myriad of conceptions and misconceptions about what these cells are. And what we're finding, and we and others, is that there's a remarkable regio-specificity to these stem cells. So if you look along the anterior-posterior axis of the gastrointestinal tract or the airways, it's very clear that there's epigenetic commitment along these, such that there's not one intestinal stem cell, but there's stem cells for different regions of the stomach, different regions of the intestine, and different regions of the colon and rectum. The other concept that we're taking tremendous advantage of is this notion of immortality. So one of the misconceptions of the field that's come much from research on fibroblasts is this notion of a Hayflick number, a limitation on the proliferative potential of these cells, of stem cells, and that they go through a crisis period and acquire mutations. That's really not the case. Adult stem cells and we're not the first to say this by any means, are intrinsically immortal. So taken out of their niche, they can grow very much like embryonic stem cells. They are intrinsically immortal. They can proliferate for an unlimited time. And the only reason that we've ever had notions of limitations on these are just because we've had poor culture conditions and they've induced a differentiation that we've attributed to senescence. The other notion that we're finding is that these are remarkably stable at the genetic level, both at single nucleotide variation and copy number variation. So we'll cover some of that. The thing that we've been very thrilled about is that they show 3D differentiation that largely recapitulates what's seen in situ. And this is something that's highly stable. So you can have these stem cells, you can grow them as immature clonogenic cells for upwards to a year. And when you differentiate them, they don't forget where they came from. And that colon stem cells will give rise to a 3D colonic epithelium that's by all measures looks like that it's seen in situ. And then finally, this I think it's important to say that these cells are not reprogrammed. So these are not differentiated cells that we've driven into a new state, this so-called reprogramming of differentiated states. We haven't seen that. We don't find that. Further, these are not organoids. Organoids are not kept in this highly immature clonogenic state. And of course, they're not embryonic stem cells nor induced pluripotent stem cells. So this notion of C, cloning and clonogenicity are key. We're able at this point to routinely, from endoscopic biopsies, generate libraries of clones derived from single cells. And this is allowing us to rapidly deconstruct the complexity of the stem cell population in those. And we call these, single cell derived lines pedigrees, but we can talk more about that as we go. And lastly, we're going to talk a little bit about disease models, how we've been able to recapitulate pretty complex pathological events such as pseudomembranous colitis secondary to C. difficile infections. How you can take a complex mixture of cells, for instance, that are the precursors to esophageal adenocarcinoma, Barrett's esophagus, and separate those into the pathogenic Barrett's stem cells and the normal squamous and gastric stem cells that they grow in, and how that's readily accomplished by this technology. And we'll touch a little bit on some ongoing work in the laboratory with regards to stem cell heterogeneity and Crohn's disease and how this technology can resolve that. And in terms of regenerative medicine, I think over the long run, this type of technology will be very powerful in that. And we've recently shown that you can use a single cell derived clone of distal airway stem cells to repopulate the lungs of mice that have been damaged with H1N1 influenza. And hopefully, with regards to the intestine, we can be doing similar things for short bowel syndrome, tufted enteropathies, and dysplastic syndromes that need some type of replacement. So, obviously on the next slide, you've got Shinya Yamanaka, who developed the induced pluripotent stem cells. And this is the dominant technology. If you work in stem cells, and I work in stem cells, this is the, much of the work that goes on in the field has come from disease models and regenerative medicine schemes that are based on embryonic stem cells and induced pluripotent stem cells. Now, there are challenges to these cells, and one, as everyone knows, they're tumorigenic if put in an undifferentiated state. There's a tremendous challenge to developing the appropriate tissues. And, the best developmental biologists in the world spend their efforts focused in on how to take an embryonic stem cell through all of the different differentiation process. So that's a challenge, one that's not faced with adult stem cells. One of the big challenges as well is there's been no evidence that you can make an adult stem cell from embryonic stem cells or induced pluripotent stem cells. Although if you drive it through a teratoma, there is showing some promise, especially with hematopoietic stem cells. And lastly, any epigenetic aspect of pathology would be lost in such cells. If you now switch to organoids and mini-guts, this has been very powerful technology. And if you work in adult stem cells, you work in organoids. And this has been developed extensively from the work of Hans Clevers and the rest. But I think there's some important notions to bring in. If you work in adult stem cells, this is what you do. So it's clearly the dominant thing. It's yielding tremendous information. But the challenges are they're typically not carried in a clonal form. So typically people make organoids from both sets of tissues. They're carried forward as these closed structures that have a low clonogenicity rate. And these sealed structures also present passaging difficulties, and they limit the access to biologicals because the apical surface is actually on the luminal side of these. So I think some of the oldest stuff is in adult stem cell cloning. People know the work of Howard Green, really the pioneer in doing epidermal stem cells. And this has been used in thousands of patients for severe burns, in autologous transplants. So what remaining skin, you clone these out and expand them for these transplants. They've been used in ocular medicine, especially for corneal epithelial transplants. And I think people have seen this very recent paper out of Michaela DeLuca's lab where they replaced the entire epidermis of a child with a blistering condition. And this is after, repairing the defect that they had. So this is very dramatic stuff, and it's been shown in a wide range of tissues. Now, all of those tissues are limited to stratified epithelia. And it's not a small number of tissues, but here's some examples of them: epidermis, oral epithelial, bladder, mammary, and prostate glands. This technology that he had was not sufficient for looking at any columnar epithelia. And this is where Xian set out to figure out how to do this. And, over a number of years, she developed this, published it in a paper in Nature in 2015. And, if you're going to be doing this technology, you should probably read this paper. This is widely applicable to a wide range of columnar epithelial tissues, and these are just some examples. I don't know why hippocampus is in here, but it's sufficient for that as well. So on the next slide, just an example of this. Just like Howard Green cloned the epidermal stem cell, this is an example of a single clone of human small intestine stem cells. There's a colony there of about 1,000 cells. Underneath that, it shows the Ki67, sort of uniform proliferation, and SOX9 is just a marker of these stem cells in the crypt. Now, early on, Wang showed that you could differentiate these in a simple air-liquid interface, akin to what people have been doing in the airways for some 40 years now. And importantly, from this single clone derived from a single cell, she was able to get a polarized epithelium and all the major cell types showing up there. And those include goblet cells, Panna cells, and enteroendocrine cells. And it was this kind of playing around that you realize that, in fact, these things are stably committed. You could grow small intestine or upper airway, and you can grow them in culture for months. And when you differentiate them, the small intestine forms a small intestine upon 3D differentiation, and the upper airway gives rise to an upper airway epithelium with goblet cells and ciliated cells. So they stayed committed. Surprisingly, at the gene expression level, these clones are very similar. They differ by just, a small set of genes, and yet when you differentiate them, they go off into their respective differentiation phase. And this is just an example of clonogenicity, and I think this is an important concept for what this system is good for. And so here you see a clone growing over 10 days, derived from a single cell. And then in two different methods, what they used was either a micropipette to select out individual clones and transfer them to 96 well plates. And the clonogenicity of these cells is very high. And you can do the same thing with FACS. But the overall numbers are approximately 50 to 80% of those individual cells are in fact clonogenic. And this, as I said, compares with organoids where it's far less than 1% in the analyses that have been done. And this clonogenicity is stable over time. And what you're looking at is passage 7 versus passage 16. Each of these are 10-day passages, so 70 days and looking at 160 days. And the good news is it doesn't increase and it doesn't decrease over time. And this is probably the most important slide in just this notion that you can take a single cell suspension, derive a library of clones from this, sample an individual clone, and because they're intrinsically immortal, you can expand these to unlimited numbers. That allows you to ultimately do parallel studies with genomics or any of these other information gathering systems, and they will all read back to that same single cell. And so this is powerful. It's a simple slide, but I think it sort of underscores the value of the system. This commitment that we talk about, we've really pushed this very far to ask how stable is this? So at passage 7, you have millions of cells derived from that single cell, and you put them in air-liquid interface, and you differentiate an epithelium. In this case, I think it's probably from the colon. If you do that same thing after 170 days, you have absolutely billions of cells from that. And again, it doesn't forget where it comes from. It forms a 3D epithelium, and we push it out to 270 days. And it's, these are, the numbers go beyond what we can count. The genetic stability through 100 days is pretty absolute. If you really push it to 250 days and do copy number variation, you can see some clones, like for instance, clone 1 and 5 are pristine, whereas you can detect copy number variations in clones 2, 3, and 4. Now, the good news is some of them are extremely stable, but they're easily monitored and mitigated. But you would never need to go to this, but it says you can. Okay. The other nice thing about doing clonal analysis for genomics is that you can you're not looking at a population where you have to do deep sequencing to figure out the spectrum of mutations. The image on the allele frequency on the right there shows that the allele frequencies are 0.5 for all these different mutations. It just makes life very easy at the clonogenic level. So on the next slide, you can see where we've gotten the entire gastrointestinal tract from single individuals and have cloned out stem cells, for instance, from the small intestines, duodenum, jejunum, and ileum. And all the clones look alike at the 2D level. And yet, even those clones show a gene signature that's repeated. So if you take two clones and you compare them side by side, there are gene sets that distinguish Duodenum from jejunum, and so on. You can do the same thing in the colon, and again, they each have a gene signature, even though they nominally look like colon from the ascending transverse and descending colon. If you look on the air-liquid interface on top of these, you can see these patterns that show up, and they're very uniform. This is all self-assembly. The duodenum showing very small images, but as you get down to the colon, you see these large whorls of cells that are incredibly uniform. By histology, you can see that the duodenum derived from a single cell pedigree. Now there's only epithelia in there, and what you see next to it is a textbook image of this, and it looks largely similar minus the stroma. And the same thing goes on in the colon. The colon looks surprisingly similar to a textbook image of colon minus the stroma and the lamina propria. So this amazed us because we were impressed that all that information is contained within the stem cell for this 3D structure. And these are just images on the next slide of just the typical markers for colon. And this pattern is repeated throughout this 3D structure. So it looks like an image if you were to image from the lumen of the colon. By gene expression analysis, again, all the stem cells for the gastrointestinal tract are largely similar, and when you differentiate them, they go off into their specialized gene expression profiles. And what we're cloning here is not unusual stuff. It has all of the typical markers that investigators in the field have attributed to columnar epithelium of the gastrointestinal tract, and you can see some of the better known ones there and which ones are high. And what you're seeing on the right is the markers typical for stratified epithelia, and of course they're different. So modeling pseudomembranous colitis. So C. difficile, as you all know, is a big problem with for diarrheal conditions. And what we have here is single cell derived colonic epithelium challenged with cloned versions of C. difficile toxin A or toxin B. And the lower images is a pathologist view of this, and so you can readily monitor this whole process occurring, and it looks very much like what goes on in vivo. And the gene expression profiles were quite informative because it's a very clean system. You're not looking at stromal input. You're not looking at input from the immune system, and so it's been very informative for what actually happens in the process of pseudomembranous colitis. So a lot of the work that we're doing now is more sort of defining the regio-specificity of the entire human gastrointestinal tract. And so we have, from these late fetal demise cases where we have a single gastrointestinal tract from one individual, we're sampling and generating libraries of stem cells, picking individual clones from those, and then differentiating them. And that's been quite informative. For the next slide, you can see that antrum gives all the features of antrum, and you can sort of march your way down. But we've actually divided the GI tract into 12 different regions. As I mentioned, these clones are regio-specific, and they're committed to those fates. So you can grow them for, upwards of a year, and when you differentiate them, they become the appropriate differentiated epithelia. So some of the work we're doing now is trying to understand that regio-specificity at the genetic level, and it's clear that the image we have here is the pattern of homeobox genes across the gastrointestinal tract, and you can see that there's a lot in the body and antrum. There's relatively few in the duodenum, jejunum, and ileum, and then it starts to increase massively again once you get into the terminal ileum, ascending colon, and down through to the rectum. So this is just the, we're able, because you can now separate all of these things, you can get tremendous resolution. And we've done epigenetic analysis for all of these things to understand why these cells are fixed in their commitment. For the next slide is an example of Barrett's esophagus. So I think people know this is a relatively common precursor lesion, and a certain percentage of these will progress on to dysplasia and adenocarcinoma. And here's just an example of what you're looking at as a white light image of the distal esophagus of a patient with Barrett's esophagus. That pink thing is the abnormal so-called Barrett's tongues, and those circles are where an endoscopist has sampled this typical 1 millimeter biopsy from these different regions. So you have normal esophagus, you can sample right from the Barrett's, and you can sample within from the gastrocardea. Again, the clones look sort of relatively indistinguishable from one another, but when you look at gene expression profiles, the esophageal stem cells look different from the Barrett stem cells that look different from the gastrocardea stem cells, and they have different markers. Now, importantly, you can now you can use the ability to differentiate these cells to clearly link the esophageal stem cells to a standard squamous epithelium that they form similar to the skin. The Barrett stem cells give rise to a intestinal metaplasia. You can see the goblet cells there, and that's without doing anything. You're not adding any CDX2, or you're not doing anything. They're just programmed to do that. And the gastrocardea cells form a columnar epithelium minus the goblet cells. So you can quickly parse through that. Looking at different clones from a lot of different patients, you can distinguish the signature, even at the transcription level, of genes that are in Barrett's esophagus compared to its closest neighbor, normal gastric epithelium. And this work, this technology has allowed us tremendous insights into the nature of Crohn's disease. So, for instance, we've got a large project going on with pediatric Crohn's, where we can sample from control patients, generate these libraries, and we can do the same thing with patients diagnosed with Crohn's. So we have two different libraries. The clones all look alike, but the fact that we can now separate them in a clonal way allows us to distinguish the Crohn's stem cells from those of the normal epithelium, and also realize that Crohn's patients have both normal terminal ileum stem cells and they have a Crohn's stem cell, and they have these stem cells have all the features of the disease. They're hyperinflammatory. They have a number of features that we think are important for the disease history. And this is just a slide comparing the lineage potential of induced pluripotent stem cells or embryonic stem cells with adult stem cells. And they're obviously, embryonic stem cells are obviously pluripotent. The adult stem cells are not. They're highly specific, and yet they're multipotent for the local epithelial differentiation. They have no lineage commitment, whereas the adult stem cells are highly committed, and they're epigenetically committed. Tremendous efforts are spent in guiding the commitment of embryonic stem cells. It's not required for adult stem cells. And in terms of the derived stemness of the final product, it's not evident in induced pluripotent stem cells, and yet the adult stem cells, that's what they do. We've used this same system, akin to Howard Green's system for cloning stem cells along the airways, and it's a very similar story. Different regions of the airways have what look like similar cells. They're all P63 positive, they're all keratin 5 positive, and they look alike in culture. And yet they have different gene expression profiles. And when you differentiate them, the upper airway stem cells, for instance, give rise to an upper airway epithelium that are ciliated cells and goblet cells, whereas the distal airway stem cells give rise to alveolar structures with type 1 and type 2 pneumocytes. And we've done tremendous work with a tremendous amount of efforts towards understanding the role of stem cells in response to severe lung damage. And in this case, what you're looking at is a mouse lung that's been treated with H1N1 influenza, which has gone in and necrotized large portions of the lung. And in those regions, you see these distal airway stem cells emerge and naturally, spontaneously form alveolar structures. And these are rare cells. Normally, upon influenza infection, they expand on the order of 400-fold, and they naturally form these structures that ultimately evolve into these so-called pod structures that ultimately evolve into functioning alveoli. And this, what we see in the mouse, is the same thing that's going on in human patients that have been infected with H1N1 influenza. And lineage tracing shows that these cells that really are there in minor levels expand and repopulate areas that have been damaged, and they assemble into alveoli. But importantly, in terms of regenerative medicine, we're able to now take cloned cells from the distal airways and transplant them into mice with severe influenza infections and repopulate these. And when they repopulate, they again form these terrific beds of alveolar epithelia. So, and it doesn't happen in cells that don't have the damage. But in this pretty much this last slide, you can see that distal airway stem cells repopulate the distal airways that have been damaged by influenza, whereas the proximal airway stem cells don't. And this, again, emphasizes the importance of cloning these types of cells for this type of regenerative medicine and the importance of this. And just in the last slide, before we take questions, we think this ability to resolve heterogeneity is going to be key as we move forward. Many of the chronic diseases we've seen are a product of heterogeneity, normal cells and pathogenic cells. And being able to separate those in a highly precise way is going to be key for both understanding the disease as well as mitigating them. We are leveraging this intrinsic immortality of adult stem cells for the ability to do genome editing and regenerative medicine. And finally, we think the platform is robust. It takes a certain amount of, you have to overcome some barriers, but once those are done, it becomes much more efficient than many of the systems that are out there. And ultimately, I think this technology will be disruptive for both understanding disease and ultimately mitigating them. So thank you. We look forward to questions.
Okay, wonderful. Thank you, Dr. McKeon, for the great presentation and the more in-depth look at how these adult stem cells can be utilized and grown and utilized for other different applications like disease modeling and regenerative medicine. So we'll move into the question and answer phase here in just a minute. I just wanted to remind the attendees that they can submit questions to Dr. McKeon and Dr. Xian here using the Ask a Question box just below the presentation screen. And at this time, I'd also like to introduce Dr. Xian, who will be joining us for the Q&A portion. So hello, Dr. Xian.
Hello.
All right.
Hello, Scott.
Thank you very much. So I'll get going. We got a couple questions in here, and the first one is for the gastrointestinal and airway model system. Have you looked into its utility to model cystic fibrosis, so like the expression of the CFTR gene, and what kind of potential do you see in the model for that?
Yeah, I mean, this is obviously an active project. Chronic airway diseases are a major goal of the laboratory. So we think it's going to be very informative. Many of these diseases, they don't know, they're inflammatory, they're pro-fibrotic, there's just a number of issues. With regards to cystic fibrosis itself, the potential for doing CRISPR-Cas9 editing is something that we've demonstrated both for back mutating the CFTR locus. And what's nice about it is because it's clonogenic, it becomes very easy to put in the guide RNAs and assure yourself that you've got a pure population. So it merges well with this type of analysis.
Okay, great. Thanks. We have another question from Christopher Heinen at UConn Health, asking if you can talk a little bit more about the isolation and preparation of these stem cells from a patient sample.
So basically, from the patient samples, we normally receive like 1 millimeter biopsy, and we use collagenase to digest the tissue and wash away the collagenase and spin down the cells, and then respond in the MimEX medium, and then grow them on top of the feeder. So the whole process is quite straightforward and robust. Normally, from 1 millimeter biopsy, we can get between 50 to 100 clones, and these clones can be picked and then passaged as single-cell derived pedigrees. So it's very robust in this MimEX system.
Great. Okay, thank you very much. Another question has come in from Wang, and he's asking if you use Matrigel instead of the Transwell system to culture 3D lung tissues.
Certainly, the 2D culture in the MimEX system, these cells can be differentiated in both Matrigel and also can differentiate in air-liquid interface. But what we found is they differentiate much better in air-liquid interface in the differentiation medium that Bio-Techne is providing now. And importantly, just like Frank mentioned, this air-liquid interface opened up these structures, so you will have an apical surface of the epithelium open to you instead of enclosed in the 3D structures, which is very important for many studies that biologists are very interested in.
Okay, thank you very much. I hope that answers that question for Wayne. Okay, so the next question is from Barry Zhao, and He's wondering if you can use the method to here to link with cancer stem cells. I know you showed how it can be used to model infection and some inflammation like Crohn's disease. But can you also use this technology to grow cancer stem cells and make cancer models?
Yes, and our unpublished data certainly support this, and they work very well. And we found the similar ratio of the clonogenic cells in tumor is around 1 out of 1,000 to 1 out of 5,000 cells. In tumor, they are clonogenic in our system. They can be propagated, and they are genetically stable, and they are tumorigenic. So the answer is yes.
Okay, great. The next question here is the MimEX system only available for human cells, and what about other species?
The MimEX system is not only for human cells, and they can be used by many other species, and they can be used for mouse, for rat, you know, and it's very broad actually.
Yeah, I mean, I think just, you know, we've used it for a number of species. Mouse is surprisingly distant. Rats, pigs, ferrets, these types of things we've readily used. We actually have less experience in the mouse. But typically, mouse is more difficult than other systems. We just don't know enough to say whether it works or doesn't work.
Yeah, but for the rat, for the ferret, and certainly it works very well. And for the mouse, and our recent data showing, you know, for example, the data from the lab yesterday showing it works quite well, and you just need to be very careful about the digestion process, and you cannot hurt the stem cells before seeding them. So there are certainly critical steps, and you need to pay much more attention to in comparison to human. But if colleagues have questions, and please feel free to email us. We are happy to address.
Great, yeah, so it sounds like so maybe the initial processing of the tissue can be a sensitive spot for dealing with different species and using it in the MimEX platform. But that's great to suggest. You have lots of work with other species, so you can help troubleshoot if other people want to try that.
Yes.
Okay, and then a question about the feeder cells. So are these feeder cells required for culturing the lung and gastrointestinal 3D cultures?
Well, so here's where I think biotech, you know, this is one of the barriers we were talking about. You know, we're using the exact same feeder system that Howard Green pioneered, Swiss 3T3 cells that are irradiated. And, you know, this is sort of a pain as far as the technology goes, but is ultimately something that Bio-Techne is providing, which is enormously helpful. So you don't have to think about it. But it does require the feeder system, as does Howard Green. Now, this recent paper by Michaela DeLuca apparently used the same system for this regenerative medicine approach on this child with blistering the skin, where they replaced the entire epidermis. So I don't know if that was a special case, but they're starting to even use, they're clearly using it in clinical cases. And there's other systems that have using these types of cells that have gotten through the FDA in the West. But it is a barrier. But once you get, you know, the fact that Bio-Techne is actually making these cells available is a huge benefit.
Okay, great, thank you. The next question, we'll just do a couple more here because we're getting close to the 1 hour time limit, but there are a few more to answer. The next question is regarding the differentiation phase. So can you speak to how long that differentiation typically takes, and does it differ between organs or even subregions within an organ?
Well, and this is a good question. And at least for GI tract, the regional stem cells are all grown in the same medium and differentiated in the same medium. And we highly recommended you seed enough immature stem cells in your transwell. And normally it takes 5 days before you can see the structures. And it takes about 10 days to fully differentiate. Then you can observe all the appropriate cell types in that particular region in the GI tract. So among all the GI tract stem cells, we observe similar time that is needed for the differentiation, and we haven't seen the big difference between the cells in the GI tract. It takes about 10 days after our liquid interface is formed.
All right. Another question here regarding, I guess it's using, taking these adult gastrointestinal stem cells or adult stem cells, and can you take those stem cells and grow in Matrigel traditional organoid structures? Have you guys done that as opposed to the air-liquid interface?
Well, we certainly tried previously, and I said if you have this very robust 2D culture, and in about 1 month you will have 1 billion cells. Almost 1 billion cells. And in the beginning for the differentiation, and we compared between the Matrigel and the air liquid interface, and for the differentiation, what we found, the cells grow very well and differentiate much better in the air liquid interface, and because they can form cell polarity better in the air liquid interface. And then the beauty, as said before, the beauty is in the air liquid interface, the apical region is open in the air. So you can, for example, for the treatment of C. diff and the others, when you measure the permeability, and it really can allow you to do this type of assays. However, it will be extremely difficult if you grow them as organoids in the Matrigel to measure, for example, the permeability, right? So certainly the air liquid interface has a lot of advantage over the organoid structures in the Matrigel, and it's easier to handle. And every time you repeat the experiments, and you always can repeat it very well, so you keep the consistency of your experiment, and also allow you to do many things you cannot do in a closed structure in the Matrigel. So this is why we decided we continue using air liquid interface for the follow-up experiments.
All right. Thank you for that answer. Okay, I think maybe just one more question here, and this one comes from me to kind of close this out. I have a background in neuroscience, and so on one of the slides you showed adult stem cells isolated from the hippocampus, or a colony of stem cells grown from the hippocampus. Just wondering if you can comment on how those stem cells behave, and how your cultural experience with them, and if they form, it's a complex cytoarchitectural makeup of that organ there. So I'm just wondering, commenting on that system in general.
Well, we're working on this. We can clearly show that we can derive neurons and glia from these cells, but I think it's just a much earlier stage of things that the kids in the lab are playing with. That's probably the best I can offer right there. they're excited about it, but they're just in this discovery phase of the utility of these things.
Yeah, that's exciting. I think that if it can do what the system's done for the lung and the gastrointestinal tissues, for the similar thing for the brain, that would be of high utility for the research community for sure. Okay, well, I really just wanted to thank everybody again who attended, and again thank our presenters today, for, Dr. Frank McKeon and Dr. Xian, for joining us and giving this insightful presentation.